It has been argued that the discovery of DNA, as well as our understanding of its structure and functioning, may well be the most important discovery of the last century. The effect of the discovery of DNA on scientific and medical progress has been enormous, whether it involves the identification of our genes that trigger major diseases or the creation and manufacturing of drugs to treat these devastating diseases. In fact, the identification of these genes and their subsequent analysis in terms of therapeutic treatment has ultimately influenced science and will continue to do so in the future. The simplest answer for why is DNA so important is that DNA is a prerequisite for life's inception. It dictates life in two manners. Firstly, it transfers hereditary information from generation to generation. Secondly, it controls the production of proteins. DNA even determines the structure of a cell, whether it would be a nerve cell or eye cell, etc. All living things on Earth have their genetic information encoded in DNA, so if it's alive and it lives on Earth, it has DNA in its cells. While very different organisms will probably have very different sets of genes encoded in their DNA, every organism uses the same code. This is why you can take a gene from one organism and put it in another and it will work, because every living thing can read the DNA code. All living things on Earth, without any known exception, store their hereditary information in the form of double-stranded molecules of DNA, long unbranched paired polymer chains formed always of the same four types of monomers, A, T, C, and G. These monomers are strung together in a long linear sequence that encodes the genetic information, just as a sequence of ones and zeros encodes the information in a computer file. We can take a piece of DNA from a human cell and insert it into a bacterium, or a piece of bacterial DNA and insert it into a human cell, and the information will be successfully read, interpreted, and copied. Using chemical methods, scientists can read out the complete sequence of monomers in any DNA molecule, extending for millions of nucleotides, and thereby decipher the hereditary information that each organism contains. DNA stands for deoxyribonucleic acid. It is special because it holds the code for every cell in your body. Every cell in your body uses DNA as an instruction manual. DNA is constructed of nucleotides in two long chains that twist around each other. That twisting shape is called a double helix. The spiral ladder has the ability to wind and unwind so that the nucleic acid chain can duplicate itself. That duplication process, called replication, happens every time a cell divides. Although it may look complicated, the DNA in a cell is really just a pattern made up of four different parts called nucleotides. Imagine a set of blocks that only has four shapes, or an alphabet that only has four letters. DNA is a long string of these blocks or letters. Each nucleotide consists of a sugar, a deoxyribose, bound on one side to a phosphate group, and bound on the other side to a nitrogenous base. There are two classes of nitrogen bases called purines, which are double-ringed structures, and pyrimidines, which are single-ringed structures. The four bases in DNA's alphabet are adenine, also known as A, which is a purine, cytosine, also known as C, which is a pyrimidine, guanine, also known as G, which is a purine, and thymine, also known as T, which is a pyrimidine. Holding the nucleotides together is a backbone made of phosphate and deoxyribose. The nucleotides are sometimes referred to as bases. Only certain sets of nucleotides can fit together. You can think of them like puzzle pieces. A only connects with T, and G only connects with C. This is known as complementary base pairing. Watson and Crick discovered that DNA has two sides, or strands, and that these strands are twisted together like a twisted ladder, the double helix. The sides of the ladder comprise the sugar-phosphate portions of adjacent nucleotides bonded together. The phosphate of one nucleotide is covalently bound, a bond in which one or more pairs of electrons are shared by two atoms, to the sugar of the next nucleotide. The hydrogen bonds between phosphates cause the DNA strand to twist. The nitrogenous bases point inward on the ladder and form pairs with bases on the other side. Each base pair is formed from two complementary nucleotides, a purine with a pyrimidine, bound together by hydrogen bonds. The base pairs in DNA are adenine with thymine and cytosine with guanine. The strands are always the same distance apart because there is always a double ring bonded to a single ring nitrogen base. The 5' end of one deoxyribose binds to the 3' end of the next deoxyribose, creating a sequence of bases that is always read in the 5' to 3' direction. However, due to DNA's anti-parallel structure, the complementary strand of DNA is flipped, so the nitrogen bases line up properly. Despite this, DNA is still always read in the 5' to 3' direction. When comparing DNA to RNA, there is a different substance attached to the carbon 2 of the sugar molecules, an H on DNA compared to an OH on RNA. Also, RNA has a uracil base instead of a thymine, as it's missing an H3C structure, and RNA, finally, is single-stranded, while DNA is double-stranded. DNA carries the information for making all of the cell's proteins. These proteins implement all of the functions of a living organism and determine the organism's characteristics. When a cell reproduces, it has to pass all of this information onto its daughter cells. This ensures survival and proper cell division, reproduction, and the passing of instructions, which govern the cellular process. Also, cells undergo mitosis and meiosis to grow or repair dead cells, and all cells must contain DNA. So for this to happen, DNA must replicate. DNA replication occurs in the cytoplasm of prokaryotes and in the nucleus of eukaryotes. Regardless of where DNA replication occurs, the basic process is the same. The structure of DNA lends itself easily to DNA replication. Each side of the double helix run in opposite or anti-parallel directions. The benefit of this structure is that it can unzip down the middle, and each side can serve as a pattern or template for the other side, called semi-conservative replication. However, DNA does not unzip entirely. It unzips in a small area called a replication fork, which then moves down the entire length of the molecule. Each parent strand serves as a template to make two new double helices. The first step in DNA replication is that the nitrogen bases in DNA must be pulled apart to create two individual strands. DNA replication begins with the unzipping of the parent molecule as the hydrogen bonds between the base pairs are broken. An enzyme called helicase unwinds the double-stranded DNA parent strand into two daughter helices, which are identical to the original DNA. Replication occurs in the 5' to 3' direction. The first step in DNA replication is the separation of the two DNA strands that make up the helix that is to be copied. DNA helicase untwists the helix at locations called replication origins, which are sequences of A and T nucleotides. The replication origin forms a Y shape and is called the replication fork. The replication fork moves down the DNA strand, usually from an internal location to the strand's end. The result is that every replication fork has a twin replication fork, moving in the opposite direction from that same internal location to the strand's opposite end. This creates a replication bubble. When the two parent strands of DNA are separated to begin replication, one strand is oriented in the 5' to 3' direction, while the other strand is oriented in the 3' to 5' direction. DNA replication, however, is inflexible. The enzyme that carries out the replication, DNA polymerase, only functions in the 5' to 3' direction. This characteristic of DNA polymerase means that the daughter strands synthesize through different methods. One, adding nucleotides one by one in the direction of the replication fork, the other able to add nucleotides only in chunks. The first strand, which replicates nucleotides one by one, is called the leading strand. The other strand, which replicates in chunks, is called the lagging strand. After the DNA has been unzipped into two single strands, primase creates short strands of RNA called primers. These are added to the DNA strand and can stick to the DNA as the RNA is a single-stranded molecule and the complementary base pairs line up. Since DNA replication moves along the parent strand in the 5' to 3' direction, replication can occur very easily on the leading strand. The nucleotides are added in the 5' to 3' direction. Triggered by RNA primase, which adds the first nucleotide to the beginning of the chain, the DNA polymerase III simply sits near the replication fork, moving as the fork does, adding nucleotides one after the other, preserving the proper anti-parallel orientation. The added nucleotides pair with the complementary nucleotides on the existing strand. So as a reminder, A with T and G with C. This sort of replication, since it involves one nucleotide being placed right after another in a series, is called continuous. The leading strand only requires one RNA primer, which is later removed by DNA polymerase I. The lagging strand replicates in small segments, called Okazaki fragments. These fragments are stretches of 100 to 200 nucleotides in humans that are synthesized in the 5' to 3' direction away from the replication fork. Yet, while each individual segment is replicated away from the replication fork, each subsequent Okazaki fragment is replicated more closely to the receding replication fork than the fragment before. These fragments are then stitched together by DNA ligase, creating a continuous strand. This type of replication is called discontinuous. One RNA primer is used for each Okazaki fragment, which is later removed by DNA polymerase I. The removal of the DNA primer leaves each daughter strand with an exposed 5' end, meaning each daughter strand is slightly shorter than the parent strand. At the ends of the daughter strands are stretches of DNA called telomeres, which protect our genetic data, make it possible for cells to divide, and hold some secrets to how we age and even get cancer. Telomeres have been compared with the plastic tips on shoelaces, called aglets, because they keep chromosome ends from fraying and sticking to each other, which would destroy or scramble an organism's genetic information. Telomeres are non-coding sequences of DNA, so if it's lost, there would be no detrimental effects. Yet each time a cell divides, the telomeres get shorter. When they get too short, the cell can no longer divide. It becomes inactive or dies. This shortening process is associated with aging, cancer, and a higher risk of death, so telomeres have been compared with a bomb fuse. As the DNA replication process is so complicated, errors do rarely occur. However, a subunit of the DNA polymerase proofreads the new DNA and removes incorrect bases. Different types of cells replicated their DNA at different rates. Some cells constantly divide, like those in your hair and fingernails and bone marrow cells. Other cells go through several rounds of cell division and stop, including specialized cells like those in your brain, muscle, and heart. Finally, some cells stop dividing, but can be induced to divide to repair injury, such as skin cells and liver cells. In cells that do not constantly divide, the cues for DNA replication and cell division come in the form of chemicals. These chemicals can come from other parts of the body, such as hormones, or from the environment.